Click for IB Biology. Here we're looking at the use of electron microscopes to produce electron micrographs. There is a blank PDF of this presentation down in the description, and also some questions so you can test your understanding. Now, as you would learn from the previous video, the capacity of the electron microscope allows us to look at objects which are measured in nanometers. There's a bit more detail to go with that. Students should be aware that they are not required to have detailed knowledge of electron microscopy techniques nor of the preparation methods. There are some fundamental differences, however, that you ought to be aware of. Instead of a beam of light, an electron microscope uses a beam of electrons. And unlike your light microscope, where you have the focusing wheels on your things, the electron microscope instead employs magnets. And these magnets are used to focus the electron beam on the specimen in order that the image can then be formed. The advantage of an electron microscope relates back to these electrons and the fact that the electrons can be thought of as traveling in short wavelength form. Light comparatively works with long wave photons. Our specimen is here, and our specimen is composed of these 5 units. But when using a light microscope, the wavelength of the light coincides with the specimen, is unable to pass between the small gaps between the specimen, and so what we observe at low resolution, which means low detail, is this solid object. But because of the short wavelength of the electrons produced by the electron microscope, they can pass between these items. And this allows us to discern them as 5 distinct objects in our image. We would use the phrase high resolution. So the advantage of an electron microscope is the short wavelengths of electrons, which allow us to produce high resolution, highly detailed structures. Now, in order to prepare the specimens for this, there are preparations of fixing and then staining. Often, always, dehydration is another process that needs to be taken before the specimen has to be cut very thinly, if I recall correctly, in the order of about 150 nanometers thickness, the slice, before it can actually be presented into the electron microscope. And this technology, these techniques are highly skilled, and electron microscopy is an entire branch of science in and of itself. So the traditional methods of preparation were created back in the late 1930s, and of course have been enhanced greatly over time. Techniques such as freeze-fracture methods. Now, in this method here, we can simply summarize this to creating very low temperatures and to actually freezing our specimen and then applying a force. You can see the force being applied here by this so-called knife structure. And what happens is that the specimen cracks along lines of least resistance, which tend to coincide with the membrane structures. And so you can see this in the image here. It's beautifully opening up the specimen between the phospholipid bilayer with the proteins showing, and we can see the results below in the electron micrograph images which are taken of this for giving us detailed knowledge of the structure down at the level of nanometers, which, remember, 1 nanometer is 1,000th of a micrometer, which in turn, of course, is 1,000th of a millimeter. So we're down at very, very small structural sizes. More recently, a technique called Cryo-EM has been introduced, and the advantage of this technique is that first of all, the beam of electrons can be much more greatly focused, much more carefully so, and so the detail that we get is better. Also, because of the methodology, which usually involves liquid nitrogen, dropping the temperatures very, very low, and this allows us to fix the specimen very, very quickly without introducing any artifacts. The elaborate methods I mentioned before are famously known to introduce features into the cells which don't actually exist. It's a consequence of the chemicals and the stains interacting with the membrane structures and creating things which don't actually exist. And so Cryo-EM allows us to get tremendous detail and is near natural condition in the specimen as possible. So we'll be finding out and seeing examples of these different forms of electron microscopy as we work our way through the course. Another technique to be aware of in terms of imaging is the use of fluorescent staining. The advantages of fluorescent staining is you can attach the light emitting molecule to very specific regions of your specimen, and it's very, very sensitive. You can introduce multiple fluorescing colors, and this allows us to see multiple events all going on at the one time in tremendous contrast. And it's compatible with a number of other techniques which exist in microscopy as well. And so you can see in the image here we have a cell in the process of division. We have the spindles here, the DNA aligning across the equator, and so a tremendous amount of detail can be derived from fluorescent staining techniques as well. Electron microscopy, microscopy itself is a tremendous skill set and a major aspect of biology in which many biologists specialize in these techniques alone. So links to the actual structures that we can see with the electron microscopy are appearing in the next video list, in the playlist, and of course the opportunity to subscribe with the channel.